Evening, everyone. We're just going to wait just a couple of minutes until our Zoom room fills up before we start tonight's seminar series. So just a couple of minutes, bear with me. Thank you. So we have a good number of our attendees now joining us. So I'd like to say a very warm welcome to everybody for joining us tonight for our seminar series. This is part 2 of our vector-borne series. And I think that we'll go ahead and get started now. So my name is Dr. Yvonne Linton, and I'm the Research Director for the Walter Reed Biosystematics Unit. I'm also the Curator of the National Mosquito Collection here at the Smithsonian Museum of Natural History. So, but today, of course, like all of you, I'm joining you from home in this COVID time. So on behalf of the museum, it's my very great pleasure to welcome you to today's program. The program today is called Ticks and the Diseases that They Carry. And it's the final program in this series, Disease Vectors of the U.S.A. So we're really happy that you could join us tonight. So for more than 2 years, the Outbreak exhibit in the museum and the associated programming have been educating people and about viruses from spillover from animals, from pandemic threats, and also from One Health. And I just want to take a moment to thank everybody, the many organizations that have supported the Outbreak exhibit, including obviously those that have provided funding, but many, many scientists who contributed their time and energies into the exhibit itself. So today's program, we're going to hear from 2 experts in ticks and the diseases that they carry to humans, but also to our pets here in USA. And after the presentation, the speakers will answer a few questions from me, and then it's your time. The audience will have a good amount of time, about 20 minutes, to put their questions forward and get them answered by the experts tonight. So if you have a look, the Q&A box is where we'll be communicating with you tonight. Throughout the program, you can post your comments and your questions there. We'll also post other links into that box as they come up in the discussion, so that you have other links that you can go to find out more. And that's where you're going to put your questions, obviously, for our speakers. So just take a moment now to locate the Q&A box, which will either be at the top or at the bottom of your screen. And you'll type your questions there, and we'll do our very best to answer as many of them as we can tonight. So we're very pleased to be able to offer this program also with captions. And to turn closed captions on for this event, just click the CC button and select Show Subtitles. You can use the same menu if you need to turn that off, and the CC button should be either at the bottom or the top of your screen. So moving on now to our first speaker for tonight, I'd like to introduce our first speaker, who is Dr. Alison Hinckley. Dr. Alison Hinckley is an epidemiologist for the Centers of Disease Control and Prevention, so that's the CDC. She holds a BS in Environmental Engineering from the University of Florida and a Ph.D. in Environmental Health / Epidemiology from Colorado State University. So in 2004, Dr. Hinckley began working at the CDC's Division of Vector-Borne Diseases in Fort Collins in Colorado. And for 3 years, she worked as an epidemiologist on arboviral diseases, including West Nile, La Crosse encephalitis, and yellow fever. For the last 13 years, her area of research has focused really onto the epidemiology of Lyme disease, of plague and tularemia, and tick-borne relapsing fevers. Since 2007, she's also worked as the principal investigator of the CDC's TickNet program, which is a collaborative public health network, which works to foster coordinated surveillance, research, education, and prevention of tick-borne diseases. Alison currently leads the epidemiology and surveillance team in the bacterial diseases branch of the CDC. So we're very, very pleased to have her today with us. And so welcome, Alison. Please, if you could give your talk for tonight.
Thank you, Yvonne, for the great introduction. I'm going to share my slides. All right, that's the hard work. So again, thank you for the introduction. My name is Alison Hinckley, and I'm delighted to talk to you this evening and a little bit about ticks and the diseases they carry. So to start with the basics, ticks are bloodsuckers, plain and simple. They need blood from animals to survive and reproduce. And when they do bite and feed animals, what's on or in them can be transferred or transmitted to their animal hosts. This is how certain viruses, bacteria, and parasites can get transmitted to people and become tick-borne diseases. Oh, a delay. So there are about 80 to 90 different types or species of ticks in the United States. Of those, only 10 or so are able to bite humans and transmit infections which cause tick-borne diseases. Here are 3 tick species that cause the most human infections: the Ixodes scapularis, or black-legged tick, Amblyomma americanum, or the Lone Star tick, and the Dermacentor variabilis, American dog tick. And I'll tell you more about these tick culprits on the following slides. So there are 15 different tick-borne diseases that we know of in the United States today. On this slide, I'm showing you 6 of the most common. Next to each disease name is one or more of the tick culprits that I just mentioned on the previous slide. This is the tick that is primarily responsible for transmitting that disease. And under each disease name is a map of the United States with a dot for every human case reported to CDC from state health departments in 2018. So from these maps, you can actually tell a lot about where ticks live and bite humans, as well as a lot about where they don't. I'm going to get into the black-legged tick a little bit more as an example. So as far as infections transmitted by black-legged ticks, there are 3 more common diseases that result. The Lyme disease, anaplasmosis, and babesiosis. And as you can see from the patterns on these maps, the ticks that are, these black-legged ticks are most abundant and most likely to bite humans in the northeastern part of the United States, mid-Atlantic, and the upper Midwest. Lyme disease has more dots on it for human cases than anaplasmosis and babesiosis, primarily because the black-legged tick is more likely to be carrying this infection. You can also see from these maps some of the dots that are not fitting the patterns quite. And in these cases, it's most likely due to people visiting or vacationing in areas where tick-borne diseases occur and then traveling back home for their diagnosis and for their doctor to report that disease to their state health department. So as far as infections transmitted by the Lone Star or American dog ticks, these can translate into the diseases for spotted fever, rickettsiosis, which includes Rocky Mountain spotted fever, transmitted by the American dog tick most commonly, ehrlichiosis, which is most often transmitted by the Lone Star tick, and tularemia, which can be transmitted by both ticks. You can see from the patterns on these maps that these ticks are most likely to bite and infect humans in the southern states and the central states. You can also see from tularemia that multiple ticks are responsible for transmitting some of these diseases. This also is true for spotted fever rickettsiosis, although those ticks are not one of the 3 culprits I just mentioned. And as a note, as you would expect, most of these tick-borne diseases occur during the summer months when ticks are most active and when people are most likely to be outside in contact with ticks. So let's find out a little bit more about the tick-borne diseases. What do they look like? Well, that depends. They can be mild to quite serious, sometimes even fatal. But in general, they can start with signs and symptoms that are common of a lot of infections. This includes fever, muscle aches, swollen lymph nodes, and a generalized feeling of illness. But there are characteristic signs or symptoms that a physician can see and use to distinguish between different tick-borne diseases as well as other infections. And let's focus in on this slide for Lyme disease. You can see in the pictures on the left a circular rash on these legs. This circular rash is called erythema migrans, or the bullseye rash. It occurs in 70 to 80% of the Lyme disease cases and is most often occurring at the site of a tick bite. When the rash is not recognized, doesn't appear, or you don't seek treatment in time, sometimes the Lyme disease bacteria can go from being in the skin and to spread to other parts of the body. When this happens, it can lead to more serious forms of Lyme disease. For example, when it affects the nerve or the nerve function, this can lead to facial palsy. There's a picture of this shown in the middle here. This is a sudden weakness in facial muscles. When it affects joints and causes swelling and inflammation, as in the picture of the knee at the bottom, this is called Lyme arthritis. And this can occur several weeks to months after original infection with the bacteria. And when it affects the heart, this is called Lyme carditis. And Lyme carditis, a picture of this is in the right-hand part of the slide. You can see a picture of heart tissue being invaded by Lyme disease bacteria. These are spirochetal bacteria, and spirochete meaning spiral. So they're little spiral bacteria encircled on that slide. And as you would guess, Lyme carditis can be quite serious, and if not recognized in time, can lead to sudden death. Nevertheless, all of these Lyme disease manifestations can be treated quite effectively with a few weeks of antibiotics, but it's important to see your provider early so you can have the best chance of having a quick recovery. So what about some of these other tick-borne diseases? There are rashes that occur for several of the tick-borne diseases. This is true for Rocky Mountain spotted fever, where we see on the foot on the left a petechial rash. This is quite characteristic of a very serious form of the disease, several days into infection, and as mentioned, is a serious sign. For tularemia, it's not uncommon to have an ulcer at the site where the infection is introduced. And for several ricketsioses, it's not uncommon to have this dark scab called an eschar that occurs. And this is the picture for Rickettsia parkeri ricketsiosis. And again, these bacterial illnesses can be treated with a few days to weeks of antibiotics. So why are we concerned about tick-borne diseases? Certainly, there's a big concern because they're increasing. On this slide, you can see the number of cases reported for each of the diseases listed to CDC from state health departments from the years 2001 to 2018. So Lyme disease cases are shown here in purple, ricketsiosis in rust, anaplasmosis in green, and blue is ehrlichiosis. And the first thing to really take in about this slide is that Lyme disease is really a very common tick-borne disease. It's the most common tick-borne disease. It's actually the most common vector-borne disease in the United States, and it's only increasing. Between 2001 and 2018, we saw a near doubling from 19,000 cases to 2017 to over 40,000 cases. You can also see that we've seen quite an increase in the other tick-borne diseases, ricketsiosis, anaplasmosis, and ehrlichiosis. The reason for this are we are becoming better at finding these diagnoses and getting them reported. Healthcare providers are more likely to be able to recognize these. And of course, more people are becoming exposed to and infected by some of those culprit ticks that I mentioned earlier. So on this slide, I mean to tell you that we're still finding important tick-borne diseases or causes of tick-borne disease. You can see that we have 18 different tick-borne pathogens, that's disease-causing germs, that cause 15 different tick-borne diseases, and those tick-borne disease names are listed here in parentheses. You can also see that there are colored bars going from each disease name up to the timeline, and those colors represent the tick that is primarily responsible for transmitting that disease. So on this slide, you can see that we've known about tick-borne diseases for over 100 years since Rocky Mountain spotted fever was recognized. More recently, we're seeing a lot more diseases transmitted by the black-legged tick and the Lone Star tick. These are the two culprit ticks. They're the most common human biters today in the United States, and it's concerning because their numbers are increasing and their range is expanding. We can also look into Lyme disease, which has a green bar around the 1980 mark. It actually was first identified in a small cluster of pediatric cases in Lyme, Connecticut in the mid-1970s, and it's grown in less than 50 years to being from a small number of cases to tens of thousands reported, as I mentioned earlier. And probably one of the most important things to recognize on this slide is the clustering of tick-borne diseases recently identified in the last 10 years. So the reasons for this are, again, we have scientific tools that are now able to find more of these diseases, but it may also be due to the simple fact that ticks are picking up new diseases from some of the animals. So why are tick-borne disease cases increasing? I've mentioned a couple of times about some of our improved tools for diagnosis and finding the tick-borne diseases, and increased clinical recognition. Here on the left, I show you one of the reference manuals developed by CDC for healthcare providers to help with diagnosis and treatment of diseases. In the middle is a major concern. It's the range expansion and tick population increases that may absolutely be responsible for much of the tick-borne disease case increase. Using the example in this figure of changes in black-legged tick distributions from a paper by Rebecca Eisen, you can see changes from the established black-legged tick populations in pink in 1996 to their expanded range in 2015, which includes all the red areas. So really expanding north, west, south, from the northeastern area, and throughout the north central part of the United States. The third reason for some of these tick-borne disease increases is a lack of highly effective prevention programs. Despite decades of science into the methods of controlling ticks, we haven't yet been able to transfer any of that science into an effective large-scale prevention program. In addition, there are not tick-borne disease vaccines currently available, although there are 2 potentially on the horizon for Lyme disease. So much of the prevention options really rely on an individual practicing it or practicing it for their family. Given that, there are still a lot of things we can do to protect ourselves from tick bites and tick-borne diseases. It's important to learn about the risks in your area. If the community you live in is one of these colored spots on one of those maps I mentioned earlier, it's important to take precaution. Protect yourself when you go outside. You can wear repellent. You can put on permethrin-treated clothing. And when you come back inside, it's very important to be able to check yourself for ticks and consider showering in order to wash off any ticks that have gone undetected. You can consider treating your pets. More and more research indicates that pet ownership is important with regard to exposure to ticks and tick-borne diseases. It's also possible for you to create tick-free zones in your yard. Smart landscaping, moving playground equipment to safer areas, and considering spraying a tick pesticide called acaricide. Lastly, if you do get exposed to ticks and you develop a fever or rash during these summer months, it's important to see your healthcare provider as promptly as possible and talk with them about your concerns regarding tick-borne diseases. So I'll leave you with this last thought. It's a little reminder about the small size and the difficulty of finding some of these ticks on your body. This is actually a picture, an image of a tweet sent out by our communications team almost 2 years ago. I'll read it to you. Ticks can be the size of a poppy seed. Can you spot all 5 ticks in this photo? And we were delighted that this went viral some time ago, although we weren't sure if it had to do with the important message or the number of amazing, funny replies regarding ruining breakfast. So with that, I'll hand it back to Yvonne and on to Dr. Ostfeld.
Thank you very much, Alison. That was a very informative talk, and I'm sure that there's going to be a lot of questions that come out there. We're going to move on to our second speaker tonight, and that second speaker is Dr. Rick Ostfeld, and he's a distinguished senior scientist at the Cary Institute of Ecosystem Studies in Millbrook in New York. He received his Ph.D. from the University of California, Berkeley, and his BA from the University of California, Santa Cruz. He's incredibly prolific and has published over 250 peer-reviewed articles. And he's written a book which is called Lyme Disease, The Ecology of a Complex System in 2011. It's published by the Oxford University Press. And he's also co-edited 5 other books. So we're very pleased that Rick could join us tonight. Rick's recent honors also include election to the American Academy of Arts and Sciences. He has a Henry J. Oosting Memorial Lectureship at Duke University. He's an elected fellow of the Ecological Society of America and elected fellow of the American Association for the Advancement of Science. Rick's research has focused on the determinants of human risk of exposure to infectious diseases, with an emphasis on Lyme disease and other vector-borne infections. So we're incredibly lucky to have Rick with us tonight. So Rick, many thanks for agreeing to talk with us, and we're very excited to see you share your slides with us tonight.
Okay, well, thank you very much, Yvonne, for that kind introduction and for inviting me to participate in this. I am assuming that I am sharing my screen right now. And unless someone shouts in my ear that I'm not, I will proceed. So Dr. Hinckley's presentation was excellent in providing a broad context for tick-borne diseases in the United States. And what I'm going to do is to tunnel into the ecology of Lyme disease risk. As she indicated very clearly, Lyme disease is the most prevalent of all the tick-borne diseases in the United States. It is the most prevalent of all the vector-borne diseases in the United States. And so the mosquito people, who some of you might have talked to, heard from earlier in an earlier program, should be embarrassed, because it's tick-borne diseases that are really the more critically important public health problem. I'm joking to some degree. Obviously, mosquito-borne diseases are very important as well. But Lyme disease is highly prevalent in the US, and as Alison indicated, it occurs in very large areas in the northeastern, mid-Atlantic states, as well as in the upper Midwest. So what my lab has been very interested in understanding is what determines human probability of exposure to the bacterium Borrelia burgdorferi that causes Lyme disease. And in order to understand what determines variable human exposure risk, we need to tunnel into the life cycle of the tick and get very nerdy. So that's what I'm about to do. I'm not sure if you can see my pointer on the screen. So I will be pointing, whether you can see it or not will be invisible to me. But so we'll start over on the left-hand side of this slide. The eggs from a generation of adult ticks hatch into the first stage, the first active stage in the life cycle of the tick. And those are the larval ticks. They emerge from those eggs in typically mid to late summer. Those larval ticks, as soon as they emerge, begin to seek a host. They only have one thing that they can eat, and that is blood. And that blood needs to come from a vertebrate host. So those larval ticks will hatch and look for a mammal or a bird typically. In some places, they will also feed from lizards as well. If they happen to find a host, they will stay attached to that host for about 3 to 5 days, doing nothing but drinking its blood and expanding in size during that period. After that 3 to 5 day meal, they then drop off the host and they begin to digest the blood meal. And so the dashed line to the right of the larval peak right there represents ticks that are sitting there on the forest floor under the leaf litter or in pores in the soil, doing pretty much nothing but digesting that huge blood meal. And they take roughly a year, almost a year to do that. They do it all winter and all through the next spring, and then they emerge as a nymph the following late spring or early summer. Again, as soon as they emerge, they look around for a host on which to feed. If they're lucky enough to find a host to feed on, and that host does not kill them in the process of grooming the ectoparasites that are on it, they will again spend about 3 to 5 days feeding on that host, after which they drop off And in this case, they spend only a few months in this state of suspended animation, this diapause on the forest floor, before emerging in the mid to late fall to emerge as an adult. And they feed for the third and final time. The adults mate while they're on the host. The females drop off and lay eggs, and they die. And then the life cycle repeats itself. So it's a 2-year life cycle with 3 different meals taken over this period of time. One very important aspect of this life cycle to note is that the larval ticks that emerge from eggs emerge free of infection with essentially any of the tick-borne pathogens that Alison mentioned in her talk. And this is particularly true of Borrelia burgdorferi, the Lyme disease spirochete. So these larval ticks are harmless at this point in their life cycle, but they will feed on almost any mammalian or avian host that they can sink their mouth parts into. It is this larval meal that determines the opportunity for the ticks to acquire an infection, which is the only way that they can become dangerous and capable of transmitting that infection onto us when they emerge almost a year later as a nymph. So, and that's what these fully red ticks are meant to represent. Those are the ticks that have acquired an infection from that host and molt into an infected nymph that can be dangerous in transmitting that infection when they feed the next year in the nymphal stage. So it turns out that these larval ticks are highly generalized in their choice of hosts. They're not picky. They feed on dozens of different mammals and birds. But it turns out that these different host species vary dramatically in the probability that they will transmit an infection to that feeding larval tick, resulting in an infected nymph that can be dangerous to us a year later. So what we've done in my lab is we've gone out and trapped and temporarily retrieved to the lab dozens of different mammals and birds and allowed the ticks to drop off, collect those ticks, allow them to molt, and then assess those newly molted nymphs for the presence of Borrelia burgdorferi, the Lyme disease spirochete. And it turns out that that creature in the lower left-hand corner is the major culprit when it comes to infecting larval ticks with the Lyme disease spirochete. This slide shows data from my lab indicating that about 90%, over 90% of the larval ticks that feed on a wild, free-ranging, white-footed mouse acquire an infection with Lyme disease spirochetes, molt into an infected nymph capable of transmitting that infection later in life. There are a couple of other species, the chipmunk and 2 species of shrews, that are sort of middling in their efficiency, what we call reservoir competence, their probability of transmitting to these initially uninfected larval ticks. But most hosts are really quite poor at transmitting an infection. And notice out to the right there, opossums, raccoons, and deer are really quite terrible at transmitting an infection to feeding ticks in the wild. What this means then is that if there are a lot of mice running around in the woods or in any sort of habitat, that means that there will be many opportunities for these newly hatched larval ticks to find one of these hosts. They actually, it turns out, are highly likely to survive when they try to feed on a white-footed mouse, in addition to being highly likely to get infected. So if lots of mice mean that there will be lots of infected ticks, we would like to be able to know what influences how many mice there are in the environment. That should be a reasonable predictor of our risk of exposure to Lyme disease, and it may pose a management intervention, that there may be something we can do to prevent that ecological risk from translating into large numbers of cases of Lyme. So I will pursue 2 angles on what influences the number of mice in the environment and how that might be used to understand and even prevent exposure to Lyme disease and other tick-borne diseases. The first of these factors is acorns. So Lyme disease occurs largely in forested landscapes in the eastern and Midwestern part of the United States. And it occurs elsewhere as well, but in the US, that's where it largely occurs. And those forests are often, though not always, dominated by oak trees. Well, oak trees have a well-known behavior, if you will, called masting. And what that means is that they unleash huge quantities of acorns, some falls, some autumns, but not others. And so this slide shows over a 20-year period in Millbrook, New York, and this is a typical pattern in oak forests of the US, that there are some years in which you might see 25 or 30 acorns per square meter of forest floor. This is averaged over the whole forest, not just under the trees. And other years in which you might see very few or even no acorns at all. What we see and the acorns are important because they are a very highly nutritious and highly cacheable or storable overwinter food supply for the mice and for other wildlife. So in a year when acorns are very abundant in the fall, they get stored over the winter. The mice survive the winter at a very high rate. They get a jump start on breeding, and they reach a peak in their abundance the summer following the good acorn fall. And we've seen that repeatedly throughout this time series. Now, if the white-footed mice achieve very high densities, sometimes a factor of 10 or even a factor of 50 higher after a good acorn year than after a poor acorn year, we expect larval ticks to have rampant access to this blood meal source to survive well and then to turn the next year into infected nymphs. And we've tested this expectation again via monitoring. We've also conducted acorn addition, mouse removal experiments down through the years. I don't have time to show you all the results. But over this 20-year period, what we see is a significant positive correlation between the acorn production in the forest in what I'm calling year T-2, so that's 2 years ago, and the average abundance of infected nymphs in the current year. So what we have determined is that acorn production is a significant leading indicator of Lyme disease risk as measured by how many infected nymphs are crawling around on the forest floor. And the nymph stage is by far the most important in transmitting Lyme infections to people. Now, there's a fair bit of variability in this graph. We know that there are other factors that influence the abundance of infected nymphs and therefore our risk of exposure. So for instance, a data point that occurred 2 years after a mast year where there were 25 or so acorns produced is much lower than our expectation. And that happened to be the driest spring over this 20-year period. And so there are weather patterns that can also influence the abundance of infected nymphs. It's not only acorns and then mice, but they have a strong and statistically significant effect. So turning to the other factor that can influence the abundance of infected nymphs, this is habitat fragmentation and destruction. And what I'm representing here is a cartoon of what happens to the vertebrate community in landscapes in which continuous forest is chopped up into smaller and smaller bits. That's what fragmentation means. So what various studies have shown, including those in my lab, is that when forest is extensive and unfragmented, you tend to experience the full complement of vertebrate animals, mammals and birds that can occur in that biogeographic area. As you chop that forest up into smaller and smaller bits, you lose some species, you lose biodiversity. But you don't lose species randomly. You lose particular species preferentially, especially larger bodied, ecologically more specialized, more carnivorous species. And what you end up with in the smallest forest fragments are primarily the small, ecologically generalized, rapidly breeding, very resilient species, in particular, white-footed mice and to some degree, eastern chipmunks. And as you may recall, these are the main reservoirs for infection with Lyme, and that also happens to be true with babesiosis and anaplasmosis as well. So one might therefore expect that these small forest fragments over on the right, dominated by the rodents, might be the places where you should expect to see the greatest abundance of infected nymph stage ticks. And indeed, in a study led by Brian Allen, we found exactly that. This is in Dutchess County in southeastern New York, where in forest fragments that were less than about 2 hectares in size, we saw significantly elevated densities of infected nymphs, whereas in somewhat larger forested areas, we saw considerably lower densities of infected nymphs and therefore risk of human exposure. So just to give you a couple of take-home messages, the evidence is fairly strong in indicating that Lyme disease risk is elevated when mouse abundance increases, and that's particularly following acorn years, but it can occur for other reasons as well. It is elevated when forests become fragmented, and I don't have a chance in this time slot to talk about climate warming. Perhaps we can address it in the Q&A, but Lyme disease risk can be elevated when cooler climates experience warming that's favorable for ticks. So two and maybe 3 of these are actionable. These scientific relationships may lead to actionable steps that we can take as residents or as public health officials. So in the case of acorn years causing mouse abundance to increase, there may be little we can do to affect the size of the acorn crop, and maybe we don't want to affect the size of the acorn crop or of the mouse population. But at the very least, acorn production, which is easy to measure, provides a leading indicator almost 2 years in advance, affording us ample time to warn people about an impending high-risk situation and to educate folks about how to protect themselves and their families or to avoid exposure to ticks. The observation that fragmented landscapes and small fragments are particularly risky suggests that we might want to avoid fragmenting forests, continuous forests, if we want to protect humans from exposure to these tick-borne diseases. And there are some places where that is certainly an important societal concern. There are other reasons to prevent forest fragmentation, and particularly when forest fragmentation is produced when we suburbanize the environment, not only are we increasing the abundance of infected ticks in the environment, we are also placing our dwellings and our yards and our activities right smack in the middle of harm's way. So this is something that is actionable in the case of tick-borne disease risk. And lastly, cooler climates experiencing warming and elevated risk is something that may be more of a future actionable item. But clearly there is little doubt that slowing the rate of climate change would help prevent not just tick-borne diseases, but various other diseases as well. In the case when tick-borne disease is very high and we can't reduce mouse abundance, we can't stop fragmentation, and we're going to be very slow in preventing the rate at which the climate warms, we are now working on a project, we meaning Felicia Keesing and I, who are the leads, and Alison Hinckley is one of our CDC colleagues who is collaborating with us on a project aimed at reducing ticks in entire residential neighborhoods. And for more on that, see that website that I've provided on this slide. And so I would like to just acknowledge the National Science Foundation and the EPA, and of course the Cary Institute for supporting this research.
Thank you very much, Rick. That's absolutely fascinating that we can actually look ahead just by the acorn yield. An incredibly complex system between, you know, the food chains that are produced in the forest. As you mentioned, the forest fragments also have an impact on which small mammals that we see there, and then also between the amount of food that's there, increasing the number of moose populations, so incredibly intertwined. And so as we think about preventing tick-borne disease, where do you think in each one of those intersections that we would have the most impact on reducing the numbers of tick-borne diseases in U.S.A?
Well, I'm a pluralist. I guess I believe in leaving no stone unturned. I think we could do a better job of managing our landscapes to prevent fragmentation, to prevent biodiversity loss. We could concentrate any necessary development in places that are already developed rather than creating new high-risk zones. I think that we can do a much better job of controlling tick populations using environmentally safe methods, and that's what the Tick Project is attempting to do. I think we can do a better job of educating the public about how to avoid tick bites, as Alison mentioned, and then work on vaccines, better diagnostic tools, better treatments. These are all very important. I would not want to rank them. I think we need to be simultaneously improving all of the above.
Yeah. I think, Alison, if I could just ask you to put on your camera as well so that we can include you and unmute yourself in this discussion. So I just want to remind the audience that we will be having a small discussion between the three of us just now regarding the presentations that we've just seen, but very soon we'll open the floor to the questions from yourselves. So please take a few minutes to think about your questions and get them down so that we can answer as many as we can today. So Alison, we're very interested in hearing about the Tick project, and Rick has alluded to it today. Could you expand a little bit more on what your objectives are for the Tick project? Will it involve purely scientists, or will it be also the general public that will be involved?
Well, I'll defer to Rick to talk about the Tick project, but I'm happy to talk about other adjunct prevention programs that have been going on as well. Maybe Rick, do you want to start?
Sure. Thank you, Alison. I'll be brief. So the Tick project takes place in Dutchess County. We are well into this project. It's a 5-year-long project where we're using 2 environmentally safe tick control interventions. One of them is a fungus that actually is effective at killing ticks. So it is a biocide. It is an entomopathogenic fungus that you can spray in people's property like you would spray a chemical tick killing agent, an acaricide. And the other is a device called a bait box, which is a small plastic box that attracts small rodents, but applies a tiny drop of fipronil, which is the active ingredient in Frontline, which we use on dogs and cats by the gazillions, and it kills ticks and other ectoparasites on them. So we're conducting this at the scale of entire neighborhoods, 24 neighborhoods. It's a fully crossed, placebo-controlled, randomized, double-blind, the scientific gold standard field effort that is probably going to kill me prematurely. But it's a hugely ambitious project, and we are bringing it to completion within about the next 12 months or so.
That's really interesting to hear of these new approaches to ticks, because it seems from the outsider's point of view that ticks seem to be so difficult to control. And I remember the very first poster that I saw in a scientific congress about ticks, which sparked my interest, was somebody showing that you could actually submerge ticks for more than a month in water and they would still be surviving. So they seem incredibly durable. Is that part of their physical makeup that makes them so difficult to be susceptible to the insecticide, or is it just that they are so widespread in the environment? Rick?
Alison, do you want to start on this one, Alison? And
I'm happy to defer that one to you as well.
well, so ticks have a relative to their size, which is quite small, they have a very thick cuticle, and that cuticle is quite impervious. And they have a very low metabolic rate. So whereas you might think that you're drowning them by placing them in water, they just don't need that much oxygen, especially during certain life stages. So they're incredibly good at escaping inhospitable conditions. It's actually quite easy to kill them in the laboratory. You can dry them out. And if the humidity gets below, 70% or so, the tick can't survive for very long. You can overheat them. It's a little harder to kill them by overcooling them. What they're really, really good at is escaping those inhospitable conditions and not needing to do very much for extremely long periods of time. They would be really good at surviving pandemic shutdowns. They would just kind of, just sit there digesting and doing nothing much else and not worry about social distancing. They don't actually social distance very well at all.
So, Alison, I was wondering, we saw a lot of the tick-borne diseases that you showed today are very much centered on the northeast of USA. Is there any reason, why there would be a concentration of the diseases there? Is it that the testing is higher there? Or in fact, is it the ecological aspects as you go across the USA that mean that you have the band where you see very few tick-borne cases?
Certainly there are different ticks in different parts of the United States. In the Northeast, there's a concentration for sure of black-legged tick diseases and definitely Lyme disease. There are a lot of people doing the things that Rick talked about, sort of moving into these fragmented areas to get more contact with the black-legged ticks. But they have high infection rates for some of these diseases there as well. And the black-legged ticks will bite humans in addition to a number of other animals, which Rick had mentioned. I mean, there are other tick species where they just don't like to bite humans, so the chance of getting an infection is less. But I may have focused on on Lyme disease or some of these other black-legged tick diseases. That's my area of work most of the time. There are certainly other tick-borne diseases of concern in other parts of the country. For example, Rocky Mountain spotted fever, which I didn't give enough credit to for its severity. It's certainly a very severe disease. It does occur commonly in the southern states, but it also is popping up in areas of Arizona that are quite arid, and in some of these areas where you might not expect them to exist. And certainly it's a concern in some of the Native American populations for being at risk. But tick species, again, transmit different types of diseases, and where the people are and how they interact with those ticks is definitely very important.
So, Alison, how concerned are you about the reports of this newly invasive tick on the East Coast? Is there any evidence that is actually involved in disease transmission for us? Or is it still too new
so this is the Asian longhorn tick, the H. longicornis tick. And this has certainly been a tick on other continents that has been responsible for transmitting some important diseases, notably some viruses. We haven't seen that here yet. And we also know that, like I just mentioned, maybe it doesn't like humans quite as much as it likes some of the other animals that it would prefer to feed upon, which is actually a great thing for us. But it's still important, regardless of what you're concerned about for tick-borne diseases. If you're in an area where you're concerned about ticks, to take precaution. You can't rely on the fact that they like dogs better than you, because sometimes you might be the only person out there. So.
That's definitely the case for me. They seem to find me very, very easily. So, so we have a lot of questions, a lot of great questions actually from our audience today. So I think we might just move on to try to see how many of these questions we can answer for them. So one of the first questions that we have, and I'll push this to you as well, Alison, is from Josephine. And the 7 and 11-year-old in Josephine's house would like to know if rolling in the fall leaves is out of the question.
You know, I don't currently live in the Northeast, so we don't have a lot of leaves for me to think about that. And Rick might have more thoughts on it, but I would tell you that leaf litter is something that you should avoid coming in contact with when it comes to ticks. In these fall months, and particularly with the black-legged tick, you're more likely to encounter the adult black-legged tick, which is quite a bit bigger than the one I showed you on the poppy seed muffin, much more likely to see. So you do have a better chance at being able to recognize the ticks in the months of October and November than you do in the earlier parts of the year. But if you can avoid it, it would be great to not roll in leaves, but you could also be very vigilant about checking for ticks. Take that shower when you come inside so that you can get all the ticks washed off that you're not able to find.
That's a great thing. And actually following on from something that you mentioned in your talk as well, you talked about in your garden how you could have a tick-free zone. And Elizabeth asked, how exactly do you create that tick-free zone in your garden?
So we talk about this in some of the resources we have online at cdc.gov/Lyme. It's about smart landscaping. It's about maybe creating rock barriers in areas so that you can provide a bit of a barrier between some of the forested areas of your property and some of the more manicured lawn. As I mentioned, sort of moving playground equipment into areas where they are less likely to have maybe more sunlit areas, so there's less likely to be ticks. There are a number of steps, and they're listed on our website, but I think, you know, a lot of folks already practice these. It's just sort of staying on top of it yearly so that you can do your best to provide protection while you're in and around your yard, which is really one of the areas where, for the black-legged tick, you're most at risk.
Thank you. I'll put this one over to Rick. Basically, Michelle has asked, what is the best way to remove a tick? And how long do they actually have to be on you to transfer any of the diseases that we talked about today?
So that's a great question, Michelle, and I'm sure that there are more people who are wondering about that pair of questions. The best way to remove a tick that is embedded in your skin, so if it's attached to you, is to move quickly. Don't waste time, don't panic, but find a pair of fine-tip tweezers, grab the tick as close to the skin as you can, and pull straight out. Don't worry too much if you get every last bit of the tick. Sometimes a tiny bit of its mouth parts might stay in your skin. You should dab it with antiseptic. You might want to consult with a healthcare provider if you're concerned about any remnant parts that are left in. But you do not want to mess around with some of the sort of folk remedies that I've heard about and heard people advocate, like putting nail polish on the tick or taking a burnt match and torturing the tick with a hot probe. Those things don't work and they waste time. And so to answer the second question, that for Lyme disease bacteria, these spirochetes, it typically takes at least a day between the time of attachment of the tick to your skin and the entry of any bacteria, the Borrelia bacteria it might have, into your body. There's some variable reports about that length of time. It has been reported as 48 hours, 36 hours. but transmission before 24 hours seems highly unlikely. Now, so that may lull people into thinking that they can take their time. It's a slow process. And indeed, for Borrelia, it is, but there are two big cautions. One is you typically don't know how long that tick has been attached when you find it. You may think you know, but you probably don't. So it could have been on you for a day, and you want to get rid of it quickly. The other admonition is that in the case of other tick-borne pathogens, transmission can occur much more quickly. So avoiding any wasting of time, getting that pair of forceps, having one handy with you when you are in the spring and summer, and even in the fall, in tick habitat, you should be ready to move quickly to get rid of it.
That's definitely a good advice to get rid of the tick as quickly as possible. Alison, Scott is asking if you can speak to optimizing your favored tick prevention for him. He's a big hiker, and he'd like to to hike in all these variable vegetations, and so he's wondering whether clothing choice or skin repellents would be your preferred method to avoid getting bitten by ticks.
That's a great question. I mean, I think it really comes down to personal preference. Wearing repellent is very helpful. Certainly, permethrin treated clothing, it can be very effective. You can wear it time and again, always have it handy. If you're a recreator, you're a hiker, you should also consider walking in the center of a trail so that you're not brushing against a lot of leaves whenever you're doing that. I wish I could prioritize for you, but I'd have to say, if you can do it all, do it all. Repellent, permethrin treated clothing, either of those would be a great way to go. But definitely, even with those precautions, it's important to conduct tick checks whenever you get home. And again, the information regarding showering as soon as you get home, really, I don't think a lot of people have heard that. It's sort of a change in messaging over these years, but it can do a lot to help you both find ticks, because there's no clothes on to stop you from finding them, but it also allows you to wash off the ticks that you're just not able to see, and that haven't attached yet.
That's definitely a good advice, and it also helps with the chiggers as well, to get rid of chiggers. So a double bonus for showering there. So one of the questions that we have here is from Doug, and he asks, is there any tick predators? Would either of you like to comment on that?
Sure, I can give that a go. We can both answer, maybe. Alison, do you want to jump in?
Now, I have heard that guinea fowl are very likely to eat ticks, but Rick, this is really your area to discuss, I think.
Okay, so yeah, so there is an older report, a scientific report about guinea fowl being effective at reducing ticks, but it was a very constrained study where guinea fowl were put in cages and ticks were put in those same cages, and the guinea fowl ate a few of them. It wasn't a very super well designed or rigorous study, and a follow-up study has shown that the effect of guinea fowl was rather weak, that they do occasionally gobble them up when they confront them, but guinea fowl tend to occupy open grassy areas and don't really enter the habitats in which ticks reach a dangerous abundance, which would be in more vegetated, shady, forested areas with leaf litter and a forest floor. We, in the tick project, we're using a naturally occurring fungus. So you may not think of a fungus as a predator, but this fungus can make its living consuming arthropods. And this particular strain of this fungus is fairly specific against ticks. There are other fungi. There are a few bacteria that can attack and kill ticks. There is even a parasitoid wasp, a tiny little wasp that can lay its eggs on nymphal and adult ticks, and those larvae that hatch out of those wasp eggs can sort of eat the ticks from the inside out. So if you want revenge against ticks, these parasitoid wasps are a great creature. But there aren't that many predators on ticks, not in this country. So it's difficult to rely on them to control tick populations. One other thing that I wanted to say is that interestingly, some of those hosts that the ticks feed on that I showed pictures of, the mammals and birds, it turns out, even though they're not going out hunting ticks, they're actually sometimes very efficient at killing the ticks that are trying to feed on them. So opossums are the champion at this. They will kill over 90% of the larval ticks that attempt to feed on them. And so a single opossum can have a rather huge effect on tick populations in the local area. So does that make them a predator? I don't know. They're just grooming, but they're really good at killing them.
And Rick, what, if any, is the effect on the animals when they are infected by the bacteria and viruses that we're talking about today?
Wild animals you're talking about?
Wild animals, yeah.
Well, we know that dogs and horses can show signs and symptoms of Borrelia infection. And there are many dog and horse owners who vaccinate their animals, and they do respond to antibiotic treatment. The wildlife that I am aware have been studied in this regard do not show any obvious disease from an infection with Borrelia. And we have studied very extensively the white-footed mouse. We vaccinated mice and asked, how does their longevity, how does their movement, how do their reproductive rates change if they're infected versus uninfected? And they don't. They also don't, they shrug off tick burdens. So there can be a mouse population where there are almost no ticks on it at all, and other mice that might have 100 to 150 ticks on them, and the mice survive equally as long, they reproduce equally as frequently. So the wildlife tend to shrug these things off. I wish I knew how they did it. Maybe there are secrets for human treatment or helping us to do a better job of fighting off either the parasite, the tick, or the pathogen.
So that brings us on to a question from Kathleen, actually. I'll put this to you, Alison. So why is there a vaccine for dogs but not for humans?
That's a good question. You know, the rules requiring approval for vaccines for humans are somewhat different than they are for dogs. And I did mention in my talk that there are actually 2 vaccines potentially on the horizon for Lyme disease, something that we can potentially look forward to in the next few years. At CDC, our role is to really better understand human vaccines, how they might be used, how effective they could be, how safe they'll be, get that information out to the public, and also to the body of scientists who make recommendations regarding use for human vaccines. So I think regardless of what exists for dogs now, there's hope that there could be this option for humans in the near future.
It seems that there's a lot of papers just now discussing the xylem, so the actual saliva of the tick, and perhaps making vaccines to tick saliva to induce your own body to fight back against any tick bite. And it seems that might be a one-stop shop. So instead of having to create vaccines for Lyme disease and for tularemia and for other pathogens, which can be quite geographically limited, it could be very neat if we could just, have a vaccine where anything that a tick can transmit to us could actually be targeted through the saliva. So I think that there's, in tick-borne diseases, it seems to me that there's a lot more activity in this field now. There's a lot more interest. I don't feel like I'm having more time outdoors just now in this COVID time, but whether we're actually more utilizing our environment, and this is where our interaction with tick-borne diseases are, or whether in fact our diagnostics are much better now than they've ever been. We have whole genome sequencing that's like $200 for a whole genome, and looking at ticks in this very open way can expose a lot of pathogens we didn't even know were there. So one of the questions I have for you, Alison, is there only one organism that causes Lyme? We have Lyme here in USA, and in Europe there's a lot of Lyme. Is it the same bacteria that's causing that in both places? Or is there an array of organisms that actually cause Lyme disease?
Well, I mean, that is a good question. There are different species that cause Lyme disease. In Europe, there are 3 or 4. In the United States, we thought we had one up until a few years ago when a second one was identified in the Minnesota, Wisconsin area. So we have Borrelia burgdorferi, which Rick has mentioned a few times, which is the predominant strain that affects, 99% of people who have Lyme disease would get that. Fairly newly recognized is Borrelia mayonii, which exists, like I said, in the upper Midwest. So they're related. They have very similar, they can be sort of identified using similar diagnostics, but there are some differences. Nevertheless, as I mentioned before, with the new Lyme disease vaccines, there is one being developed in Europe right now that would cover all the strains of Europe as well as those in the United States. And so that future vaccine is expected to be marketed and available in both areas. So quite a bit of coverage.
That's super hopeful for the future. I think that Lyme disease is one of those very debilitating diseases, and it seems that it's incredibly difficult to diagnose as well. There's a lot of people that you meet who say, I had Lyme disease for 2 years and the doctors couldn't diagnose it. Do you want to expand just a little bit, Rick, as to why there's such a problem with the diagnosis of Lyme disease in human patients?
Well, I'll say a little bit, but I might defer this one to Alison for more extensive comments. I think one of the problems is that the diagnostic tests currently in use are based on antibody detection. So they're serologic tests. And it takes a little while for our bodies to produce detectable levels of antibodies following our exposure to the Lyme disease bacterium. So if someone is in the early stages of disease, they are in that bullseye rash phase, for instance, they were exposed only a week or 2 ago, and they get a blood draw to do a confirmatory diagnosis, it may come back negative, but only because there's been insufficient time for that antibody response to be detectable. So that can be it's not a false negative in the strict sense, but if one were to wait another couple of weeks or a week, that exposure would probably be more detectable. There are rates of false negatives and false positives that I think are widely thought to be inadequate. We can do a better job than we currently do, but it is proving to be difficult to assess through these serologic methods. Yet on the other hand, directly detecting the bacterium using something like PCR, so detecting the nucleic acid of the bacterium, is very difficult because it tends to leave the bloodstream and enter other tissues where it can occur in very low numbers and be very elusive and hard to find. So there are multiple reasons why we have some struggles. And I'll stop there.
So it seems that the bullseye rash is a very, very good indicator for people to know that if you have this bullseye rash, then you should really seek some medical help. Maybe Alison, you can answer this question from Heather. She's in Oklahoma, and she says, tick experts say that we have no Lyme disease in Oklahoma, yet we do have tick bites that cause a bullseye rash. Do any of the other vector-borne diseases that you've talked about today cause bullseye rashes, or is this most likely to be stary?
Well, that's probably the answer right there. Star eye, which is southern tick-associated rash illness, which occurs, looks indistinguishable from the EM rash, the erythema migrans rash that I showed pictures of. This can happen in some people after the bite of the Amblyomma americanum, the Lone Star tick, which Oklahoma is famous for having an abundance of. So in the case of star eye, there's no known infectious agent. People looked for it, I know at CDC, for over a decade, and it's thought to be an immune response, possibly to the tick saliva. So I think, you know, what's important for folks to know on the webinar is, you know, there's certain criteria for getting Lyme disease. Number one, you have to be in an area where the black-legged tick is. And number two, that black-legged tick has to have the infection. And number three, it has to, you know, find a way to bite you, which they're not that fast moving. Find a way to bite you and be attached, as Rick said, for at least 24 hours. So outside of those areas on the maps that I showed for black-legged tick diseases, it's pretty rare to have Lyme disease, but there are other tick-borne diseases to be aware of and take precaution against.
Well, we are almost out of time for today's seminar, so I want to really reiterate our thanks to our speakers today, Alison Hinckley and Rick Ostfeld, and to our audience. We're so incredibly glad that you could join us tonight for this really lively discussion on the disease vectors, particularly ticks today in the U.S.A. We thank our speakers for their amazing presentations, very, very interesting discussion topics that we went through, and also to the audience for their insightful questions. I hope that you all learned a lot today and that you'll return for more webinars through the Smithsonian in future.